Hello everyone. I'm very thankful for this online conference so that I could present my results. Now let's go to my talk, which is integrated transcriptomic and metabolic analysis of garlic shoot tips, which provide insights into cryostress conditions. Garlic is an annual herbaceous plant. It is native to Central Asia, which is grown for over 5,000 years ago. It is first domesticated in the central mountainous region of Kyrgyzstan and Kazakhstan. Very recent research pinpoints that the likely center origin of garlic is near the Tian Shan mountains. The top commercial producers of garlic are China, South Korea, India, Spain, and the United States. Europe has a long tradition of utilization because of the Roman legions. As they conquered the northern Europe, they started introducing garlic into the Europe. And here, because of the cultural and climatic diversity, the diversity is very high. Allium species have a very large genome sizes of 10 to 20 gigabase pairs. They are larger than the eukaryotes. Garlic is diploid and its nuclear genome is estimated to be at 15.9 gigabase pairs, which is slightly smaller than onion and 32 times larger than the genome of rice. Hence, it requires de novo transcriptome assembly as it is not fully sequenced. This de novo transcriptomes assembles short nucleotide sequences into longer ones without the use of the reference genome. Garlic is sterile. It lost its ability to form seeds in the course of its domestication. As we all know, for many centuries, the garlic is clonally propagated, which has resulted in the bottleneck effect of genetic variation. Field maintenance of garlic is difficult because of the abiotic and biotic factors, and each year susceptible accessions are disappearing. In IPK, 424 growing accessions are maintained in the field, and we have 159 accessions which are duplicated in cryo. Now we will see the garlic cryopreservation protocol. Cryopreservation is a multi-step process. The shoot epices excised from the bulbils are pre-cultured on 10% sucrose medium overnight. They are then treated with the loading solution and dehydrated by plant vitrification solution 3. Later, they are freezed and then rewarming takes place. After rewarming, we wash with the washing solution to remove the traces of the cryoprotectants, and finally plated onto the medium to access the survival and regrowth percentages. During the course of the cryopreservation, we see declining regrowth. These can be the factors of physiological, morphological, biochemical, and molecular factors on encountering the stress. Now with this, I have taken few objectives of my research. That is, how does the survival and regrowth differ between genotypes after cryopreservation? How are the transcriptomes altered during cryosteps, and which GO terms are enriched? Which ultrastructural changes are observed during cryopreservation? Finally, how does the energy charge affect the metabolism of cells? To study the first aim, I have taken two different genotypes, allium 877 and 524. So after cryopreservation, the survival of allium 877 is around 97%, and when we see with the allium 524, it is around 69%. So we see here a decrease in allium 524. When we see the regrowth, the allium 877 as little bit is increased in comparison to that of the allium 524. From this, we can say that the allium 877 has a better survival in comparison to that of the allium 524. Now let's see the stepwise comparisons during cryopreservation. The fresh, here we see the after dehydration, the survival is reduced in allium 524 in comparison to that of the 877. This clearly indicates that this accession shows a clear less survival. Now the shoot tips which have been processed after cryopreservation have been given for the Illumina sequencing. The raw reads obtained from the Illumina sequencing are used check for the quality control with fast QC, and they are trimmed. The raw reads are trimmed with the cut adapt. Finally, we have the high quality reads which are then assembled with the Trinity assembler. And now we have the de novo assembly transcriptome. To analyze the gene abundance estimation, we use the Callisto method. And finally, the differential expression of genes is studied with EDGE-R bioconductor package. And final set of unigenes are functionally annotated. Now let's see how many genes are differentially expressed among the garlic accessions and in cryosteps. Here, allium 877 has around 5,039 total transcripts. Of these, 2,686 transcripts are differentially expressed. In allium 524, 5,329 transcripts are the total in number, and the differentially expressed are 2,786. Here we do not see the big differences. Now let's see the principal component analysis of both the genotypes when we see the three steps. Here we see the allium 877 accession which showed the better survival. The treatments are well separated together, fresh, dehydration and liquid nitrogen. And the other principal component for the 524 which indicated lower survival is also well separated with the treatments. Now let's go to the genes which are expressed in all cryosteps. Allium 877 has around 111 genes which are differentially expressed. Allium 524 had around 193 genes differentially expressed. Among both of the accessions, 4 set of genes were differentially expressed. Now let's see with the heat map what are these 4 genes and how they are expressed. They are inorganic pyrophosphate, expansin genes, histone complex and catalase. When we see in comparison to fresh, all the genes are strongly upregulated after liquid nitrogen treatment. So with this we can say that the expansin and catalase are coding for the oxidative stress response, whereas the histone complex is coding for the DNA methylation. Now we will see the enrichment analysis of allium 524 and 877 after liquid nitrogen treatment. There are specific GO terms which are enriched. The first one is the integral component of membrane. This GO term is strongly enriched in allium 524 to that of 877. The second one is the ATP binding. ATP binding seems to be less enriched in 524, and oxidation reduction process is more enriched. Now let's go to the 2 GO terms on further view. This integral component of membrane we will see in the ultrastructural study. So here we see the ultrastructural study of allium 524. Here is the fresh apical shoot tip and the sections of the fresh shoot tip after transmission viewed by transmission electron microscopy. The nucleus and all organelles seems to be clear. So after the pre-culture treatment, there again the cell organelles do not change. We do not see any alterations in comparison with fresh. And to the dehydration, few changes can be seen that the nucleus are converged together to heterochromatin nucleus, and the vacuole is getting bigger. So loss of dictyosomes and enlargement of vacuoles are noted. After liquid nitrogen treatment, in the overall of the apical shoot tip, we see a large vacuolar space. This can be that vacuole are enlarging in size, and after freezing treatment, the nucleus is converged. Now let's see what are the genes are responsible for this regulation of cell wall and cell organelles, which are differentially expressed. There are few genes which are responsible for the cell wall. They are pectin methyl esterase and expansin, which are upregulated, and xanthine genes, which are downregulated. There are also a set of genes. There are also genes for vacuole, which are synthesizing gene DMP1 and chloroplast genes. They all are upregulated. And we also see the peroxisome genes are upregulated. And final set of genes are the mitochondrial genes, which are upregulated from RM41A to TRXH4. And the other set of genes, kata, pump, and heat shock proteins, are all downregulated after liquid nitrogen treatment. As we all know, this mitochondria is the powerhouse of the cell where the ATP production takes place. So when we see here the differences of the differential expression, let's see what happens to the ATP. Now let's see the second go term, ATP binding proteins. There are few ATP genes which might also be involved in the production of ATP, which shows the changes also in the mitochondria. These genes are responsible for ATPase activity, and the ACLA and ALA are responsible for the transport of phospholipids or the synthesis of cytosolic acetyl coenzyme A. The next set of genes are the plasma membrane ATPase, which are responsible for transport from endoplasmic reticulum to Golgi apparatus. And the next set of genes is the ATP-dependent degradation of ubiquitinated protein. There are also putative dead box ATP-dependent RNA helicase 29 genes. And final set of genes are vacuolar ATPase, which are responsible for acidifying variety of intracellular components. Now, among them, we also have differentially regulated ATPase genes. This ATPase can also be the reason for the reduced metabolic activity. We can see either the ATP is decreased or how the changes happen. To see this, we also performed ATP analysis, and here during dehydration, this ATP is decreased, and this decrease is significant. So we know the ATP amount is getting lower. The production of ATP is less. What happens to the energy charge? So to know the energy charge, we also need to do adenosine monophosphate and adenosine diphosphate. When we see a decrease in the adenosine diphosphate during dehydration and increase in adenosine monophosphate, the energy charge is calculated, and there again we see the decrease during dehydration. This decrease is significant. So we clearly know the ATP is getting lower and the energy charge is affected during dehydration. So with this, I would like to summarize that the reduced regrowth percentage we have observed in garlic cryopreservation among 2 garlic accessions, and ultrastructural changes also showed dehydration and liquid nitrogen steps which emphasize the occurrence of cryostress. And some genes related to cell wall and organelles under cryostress were also differentially expressed. Finally, energy charge reduced during dehydration step, and we also saw some ATPase genes which are significantly changed. With this, I would like to acknowledge my team and my supervisor, Manuela Nagel, and thank you for your attention.